Thank you so much. So a quick overview. First of all, my name is Tina Shepard. I work for Praxis Medical. Praxis is a US company located out of Tampa, Florida. I cover all of the international markets. We manufacture a first motorized fine needle biopsy device. We received FDA July 2020, so right in the beginning stages of the pandemic. We received CE approval the following year, April of last year. Currently, we have distributors covering the US, and then for international, we have so far 18 distributors covering about 38 countries. We are seeking distributor partners for mainly the Nordics, the European or the Eastern European countries, Baltics, Poland, Romania, countries like that. But we do have some coverage mainly in Western European countries. So that's just a quick overview of the company. Let me just share my screen.
We can see it.
Okay. So CytoCore, like I said, is the first motorized rotating fine needle biopsy device. So biopsy, if everybody understands biopsy correctly, that it's used to diagnose obviously suspicious lumps and determine whether or not it's benign or cancerous. So far, the choices that the doctors have are three choices. You've got the open surgical biopsy, or then you've got a lesser invasive device, a core needle biopsy device, or you've got fine needle aspiration. Those are the three choices that the doctors have. Typically, they're going to choose either a core needle biopsy or a fine needle aspirate. The difference are what we're trying to provide is there's a gap between core needle biopsy and fine needle aspiration. And so the whole role for CytoCore is basically to capture more material in fewer passes and having better clinical outcomes for the patients and making it, minimizing the patient's risk. So the solution, like I said, with core needle biopsy, first of all, let me just go back to core needle biopsy. Obviously, open surgical biopsy is something that the doctor is going to use as a last resort. Core needle biopsy, it's less invasive, obviously, than surgical biopsy. The difficulty with core biopsy needles is a larger gauge size, and there's a risk associated with larger gauge size needle. And then the other choice is fine needle aspiration. It's a very small needle. It's just attached to a syringe. The problem with fine needle aspiration is there's a lot of frustration. There's a lot of problems trying to capture more material or cellular material. And it takes multiple passes to be able to capture enough sample material. They would love to be able to use a smaller needle to be able to get enough sample material to give to the pathologist to make an accurate diagnosis, but that's just not the case. And so they have to automatically choose a core needle to be able to get enough material to give to pathology to make an accurate diagnosis. So where CytoCore comes into play is basically bridging that gap, like I said earlier, bridging that gap between core needle biopsy and fine needle aspiration. In a perfect world, the physician would love to be able to use the smaller gauge size needle to be able to get enough material, but that's just not the case, because usually the smaller the needle, the more passes they take, as a longer procedure. And then there's a high non-diagnostic rate that's associated with fine needle aspiration, meaning that that patient has to come back for a repeat procedure. So our primary targets, first of all, CytoCore is intended or approved for any soft tissue. We target typically thyroids because thyroids, the current technique is fine needle aspiration. but there is a high frustration level with performing thyroid procedures. There's a high non-diagnostic rate. The statistics show there's maybe up to about a 30% of non-diagnostics. And it all depends on the patient's or the end user's skill set when it comes to performing FNAs. So about up to 30% is non-diagnostic. Those patients have to have a repeat procedure. Out of those repeat procedures, those patients go on, about 50% of those patients go on to have a surgical intervention. And out of those 50% patients, 75% of those patients is benign. So we're trying to avoid having an unnecessary surgical procedure. We definitely want to reduce the amount of repeat procedures. And so that's the whole goal behind CytoCore, is to be able to reduce the non-diagnostic rate, and then make it easier for the patient. And obviously the end goal is to be able to give more material to the pathologist to make an easier diagnosis with. Lymph nodes is another target that we go after. Lymph nodes is typically cored instead of doing an FNA. Lymph nodes are very difficult to core. It has a higher cancer rate, about 55% in lymph node versus about 5 to 10% in thyroids, where it requires more tissue acquisition. So we like to go after lymph nodes and give the doctor a safe alternative to core biopsy, where they can use a smaller gauge size needle to be able to get enough material in 1 to 2 passes instead of multiple passes. And then lung biopsy is another typical target that we go after, just because they core for the lung biopsies, they typically core. But the problem that's for lung biopsies is that it's associated with a lot of risk. Main risk is pneumothorax. So if we can prevent a pneumothorax, give that doctor, again, another minimally invasive, safe alternative option instead of doing a core, we can give them CytoCore that's going to offer them the ability to use a smaller gauge size needle and still capture enough material just like they would with a core biopsy needle. So CytoCore results, if you can notice this image right here, this is a thyroid standard FNA. So the criteria for adequacy is defined 6 groups of follicular cells, each containing about 10 cells. So this is an image of a first pass doing standard FNA for thyroid. If you can see this image compared to they did the second pass with CytoCore, as you can see, a lot more material compared to standard FNA. We have some clinical data. We have a hospital in Florida called Baycare, where their non-diagnostic rates, they did about 250 cases, and the majority of the patients only required 1 pass for adequacy. And their non-diagnostic rate they determined was around 20 to 25%. So after conducting this study, they were determined that they were able to reduce their non-diagnostic rate to about 3%. So that was really significant for this particular hospital, because they did realize that they had a high non-diagnostic rate in their facility. And so once they started using CytoCore, they were able to not only reduce the amount of passes that was required, they were taking a minimum of 3 passes, depending on the physician. It was either 3 passes, but some of the physicians were taking 6 to 8 cases, or 6 passes. So for this particular hospital, the interventional radiologists were performing all the FNAs. Once they brought on CytoCore, they were able to give the instead of the radiologist performing these procedures, they handed it off to a physician assistant that started doing all the procedures. And the procedure time was about 45 minutes, and they were able to reduce the procedure time down to about 10 minutes. So not only they took it away from the interventional radiologist performing the procedure to a physician assistant, 45 minutes they were taking to do a procedure previous down to about 10 minutes. One pass, 2 passes max instead of multiple passes. We have a hospital in New York that compared CytoCore to their core needle biopsy. They were able to determine that it was less trauma to surrounding tissue. We also have a white paper that shows that there's less risk of bleeding that's associated with CytoCore. And then overall, we've been able to determine get some really good positive clinical feedback where the physicians are reporting that is a much better positive experience when they use CytoCore. All right, a quick little demonstration of how it works. So you can do it under imaging, ultrasound imaging guidance, or in CT. And if you'll notice, so the same components, if you're familiar with fine needle aspiration, it's just a small needle attached to a syringe. So with our device, what it's doing, you can take the same components of just a small needle, 22 gauge to a small 25 gauge is compatible with our device, and then attach a standard 5 ml syringe to our device. Now it's one single disposable unit. It's like it's showing under ultrasound here, but it's one-handed, meaning they don't have to use 2 hands to pull the syringe back to try and activate the aspirate. And you can use one-handed design to create suction as well as activating the motor. So standard technique, and this is rotating the motor, is oscillating backwards and forwards. So it's basically turning a standard needle into like a little mini drill to capture a lot more material in fewer passes. So standard, they're just collecting minimal sample material, versus with CytoCore, once you're rotating that needle, as you can see here, it's going to capture a lot more specimen in fewer passes. And again, here's just a quick side-by-side comparison. Any questions?
Thank you, Tina, for the presentation. I'm sure there will be some questions in the audience. So now the floor is open to questions.
Hello, Nina. My name is Carlos Borges. I'm from Brazil.
Hi.
Hi, how are you? A few questions for you. First of all, do you have a distributor in Brazil?
That's not my market, but I don't think we do.
Okay.
I think that's open.
Okay, just a question. What is the actual price difference you have from your device to the regular biopsy device? Is that a huge price difference?
There's a big difference between, if you're going to compare it to FNH, just a small needle, it costs like nothing. But so we typically price it or state that it's similar to a premium core needle biopsy.
Okay, that represent, because I'm not in the biopsy field, but I'm on the oncology field, selling diagnostic tests. And I do understand the difficulty, for example, on lung biopsy to collect material. Right. So lots of time we have, I mean, this could be very important for the patient and for the pathologist to be able to collect the material in fewer passes or quicker and make sure he has material to work with. So obviously, I could understand that would be more costly. I know the technology behind it, but anyhow, I just want to see it's twice the price, 3 times the price, 4 times the price.
Yes, it's much more expensive. I mean, it's a motorized device. So if you can imagine, just to manufacture it, there's a lot more components that's involved to make it a motorized device. There's a battery, it's battery operated. So the battery is housed in the device. And so it's a little bit more, obviously it's a little bit more expensive to produce. But the value add is really comes down to what the pathologist is saying. And what we're hearing from pathology is not only are they getting more material, but they've got better quality material to be able to go on and do additional testing. And so a lot of times they just don't even get, number one, enough material to even make an accurate diagnosis the first time around. So it kind of goes back to the statistics of the higher rate of non-diagnostics, right? So they've got that limitation. So what we're hearing again from about CytoCore is not only are they getting more material so they can now make an accurate diagnosis, but hey, they also have additional material to do more ancillary testing like cell blocks and things like that. And so what we're hearing from pathology is that, you know, doctors will say, okay, well, I don't want to do FNA because I just don't get good samples. So I'm going to only do core. But the limitation with core biopsies, they're giving you really good tissue samples, but it also limits pathology from doing additional testing. So what we're hearing from pathology is they like to have the best of both worlds, meaning they would love to be able to have cellular material to do their cytology analysis, but they would also like to have tissue fragments to be able to do a histology analysis as well. And that's where CytoCore comes into play, is that we're giving the best of both worlds, meaning we're giving them cellular material from the aspirate, and we're also giving them little microcores to be able to do a histology analysis as well. So that's what's really important to the pathologists, and that's who's really helping to get it into a lot more hospital systems to get their buy-in. Not only just the doctor that's performing the procedure. I mean, so it's two things. So you're giving a really good tool for the physician to be able to perform the biopsy, right? We're making it easier for them to perform. There is less procedure time, and it's also much better for the patient. But the end goal is to be able to give, you know, a really good sample to the pathologist, so we can make their life a lot easier and give them more ways to do some more ancillary testing as well.
I got you. Nice. If you could send me some, probably have on the presentation here, but if you could send me a contact to see if Brazil is represented, I'm going to do a little bit more of a research on it. Thank you very much for your time.
We will manage the connection and share the details you need, Carlos. So for sure, we will follow up. I can also see one more question in the chat from Yvonne. She's taking you for the presentation, Tina. And so now I just read the question. I do not remember if there was an open opportunity in the Nordic countries. Is the region already covered?
For the Nordics, we currently have a distributor that is out of Denmark, but there was also covering the Nordics, but we might have a restructure. And so Norway, Sweden, and Finland may be becoming available soon.
All right, thank you. I see Yiri has a question.
Yeah, thanks. Tina, thanks a lot. Great presentation. And on behalf of the Aspironics for Czech, Slovak, Poland, we are in the biopsy business. We are in breast care biopsies in multiple areas. So I'm definitely keen to talk more specifically for distribution in our region. So thanks a lot.
Yeah, looking forward to it.